So today we are going to discuss about the pseudogenes. What are pseudogenes? So pseudogenes are the gene copies that have coding sequence deficiencies like frame shift and premature stop codons but resemble functional genes. For example, pseudogenes basically, if suppose this is a gene, if this is duplicated, this gene is forming one more gene like A. But during the formation of duplication, what happened? There are certain mutations. Because of this, that particular gene becomes non-functional, or because of some premature termination codon, stop codon. So even though the copy of this A gene is present, but then also it is non-functional. So this is basically the definition of pseudogenes. So coming to uh, the definition. So pseudogenes are gene copies that have coding sequence deficiency like frame shift mutation or there are any premature stop codons. Then these genes becomes non-functional and are known as the pseudogenes. Now the first pseudogene was reported for 5S DNA of Xenopus. Xenopus is an amphibian. It looks like a toad. Coding for the oocyte type 5S RNA in 1977. So currently approximately 20,000 pseudogenes are estimated, which is comparable to the number of protein coding genes in humans. Like I said before, pseudogenes, they are originating from the decay of genes that originated from duplication through evolution. So first of all, there is a duplication of the gene, and after that duplication or during the duplication process, there is a decay of genes, and that decay of genes is because of the point mutation, either because of the insertion, deletion, misplaced stop codon, or frame shift of a gene. And because of these mutations, the gene copy that is present that becomes non-functional, and it is known as the pseudogenes. The decay may occur during duplication, and it may lead to the loss of a gene function, loss of productivity, expression of RNA protein coding ability results in the production of pseudogenes. So basically, they are not functional theoretically. So there are two types of pseudogenes. One is the unprocessed and another one is the processed. So unprocessed again are of two types, unitary and the duplicated. Unitary formation of the non-duplicated means there is no duplication. Just remember this point, they are non-duplicated. The unitary type of unprocessed pseudogenes, as their name is suggesting, they are unprocessed and there is no duplication, then they are unitary. And because of this, a single copy parent gene becomes non-functional. And the very good example of this is the gulo gene in the humans, that is a unitary pseudogene, and that is the reason humans cannot synthesize vitamin C because the only gulo gene can that is present in the human beings that become inactive. And because this gene become inactive, it is known as the pseudogene. Now coming to the second one, which is duplicated, means the gene is duplicated and then that become non-functional. So unprocessed and the duplicate pseudogenes keep their intron exon structure, for example, many olfactory receptor pseudogenes. So if we see the differences, you can see in case of the unitary pseudogene, there is only one copy of the gene and that becomes non-functional, and that is why it is known as a pseudogene, and this is the unitary. So loss of the function of the one copy. In case of the duplicated, as their name is suggesting, uh, they are duplicated, so there are extra copies present, but that copy is not functional. So functional gene exists? No, it is here because this is duplicated. Has introns? Yes, both of them have introns, and mechanism is, in case of the unitary, it is gene disruption. In case of the duplicated, is that during the process of gene duplication, there is a mutation, either insertion or deletion, and because of this, they become non-functional. So the good example of the unitary pseudogene is the human pseudogene, or for the and the example of the duplicated unprocessed pseudogene is the many olfactory receptor pseudogenes. Now coming to the processed pseudogenes. So what happened in the case of processed pseudogenes? They are forming the mRNA. mRNA is forming the cDNA. cDNA is inserted into the genome and then that becomes non-functional. So the processed pseudogenes lack introns, 5' protose sequences, and have flanking direct repeats and 3' poly A tail because they are like uh, they are they are being formed because of the transcription process. You can see first the mRNA is formed, then the mRNA is reverse transcribed, then the cDNA is formed, and then they are inserted into this. So processed pseudogenes are formed through retrotransposition. Retrotransposition occurs by reintegration of the cDNA into the genome at a new location. Okay. So unprocessed means there is no processing, there is no RNA, no cDNA, nothing is formed. And because of that, they have introns. In case of the processed pseudogenes, they are processed. So there is RNA, cDNA is formed. So they lack the introns. But they have the poly A tail. So you can see the duplicated processed pseudogenes and processed pseudogenes. What are the differences? This is gene duplication. This is mRNA reverse transcription. Now introns in duplicated unprocessed they are present because they are unprocessed. In case of the processed they are absent. Promoter usually present, usually missing in case of the processed pseudogenes. Poly A sequences no. In case of the processed, yes. Location near the parent gene. Processed it can be occur at any site or random genomic location. Parental gene functional parent remains. Processed in this also parental gene is still functional. The structure is multi-exon. In this case, this is single exon. Why do organisms keep pseudogenes? The extra copies of the functional gene accumulate mutations and this maintains original gene function. Gene duplication may give rise to entirely a new gene with completely different function, may be good or bad. Recent papers indicate that some pseudogenes exhibit functional roles such as gene expression and gene regulation. It is not the case that they are always non-functional. They could be functional also, as the new research is indicating. And genetic code of the organism can be duplicated by copying errors. So during the process of duplication, there are errors, and these duplicated genes will be passed from generation to generation. Since pseudogenes accumulate mutations over years, it's not just you know in in a short period of time. Number of mutations of these called so-called fossil molecule provide an estimate of their age. So for the measurement of the age also, these pseudogenes are sometimes used. So what happened whenever there is a process of duplication, there are errors, but the frequency or the rate of these errors are not very high. And because of that, you can estimate the age of the fossils. So mechanism of the action, if you talk about, so for example, antisense pseudogene transcript, just like the siRNA. It can combine with the sense genic transcript, and this regulates functional gene expression level. Why? How come? Because if this antisense sequence is binding to this sense sequence, this sense sequence cannot form the protein, and that's how they can regulate the functional level. Similarly, folded pseudogenes transcript from the hairpin-like structure to form siRNA, again the same thing, and these siRNA repress the gene function, gene expression. Then you can see the third one is the coupled miRNA regulate both the gene P10 and the pseudogene P10P1, which is a pseudogene. So P10P1 pseudogene binds to this miRNA and reduce the expression of this miRNA. This allows P10 to escape from miRNA expression level. For example, if you see, this is the gene P10. This is the pseudogene P10P1. miRNA is binding to this, so it is losing its ability to binding to this, and because of this P10 can express and it can escape from the miRNA repression regulation because miRNA is busy in binding to the P10 pseudogene. So that's all about. And yes, one more thing. There are different methods developed by the independent groups to identify pseudogenes. You can Google it, regexp, pseudopipe, pseudofinder, retrofinder, and gspat. These are the some tools which are used to find to identify the pseudogenes. So that's all about today's lecture. Thank you so much.